course. Okay,
Sure.
so we're really excited to have Dr. Damjanovski here to provide us a talk. So just a little background, Dr. Damjanovski was actually on my PhD defense and asked a lot of challenging
Sure.
questions. So I'm glad that the tables are now turned and I can I can hopefully ask some as well at the end of his talk. He's an associate professor in the Department of Biology at Western, also associate scientist with the Children's Health Research Institute at the London Regional Cancer Program. He did his postdoc at the NIH in Maryland and PhD at the University of Toronto in the Department of Zoology with Dr. Maurice Ringuet. So historically, his research program has used the model organism Xenopus to study the developmental roles of extracellular matrix proteins. And his work has now evolved to include other cell lines and model systems to better understand the roles of ECM proteins in development and disease. However, as if you were listening to our conversation before the talk today, his research has taken a little bit of a tangent, and he's going to talk today about more recent work where he is investigating developmental plasticity and thermal performance and cardiac function in Atlantic salmon. So with that, I'll turn it over to Dr. Damjanovski, and yeah, I look forward to hearing about your lab's recent work.
Thanks, Rob. I assume I'm sharing my screen okay? It's all good? All right. So thanks for the invitation. I would have loved to be there. Instead, I'm looking at a little picture of myself, which is actually quite disheartening. But regardless, as Rob just said, I'm going to be talking about sort of work that's a little tangential to me. I'm sort of a developmental biologist, molecular biologist by training. And the work I'm going to be presenting today is largely the work of a talented PhD student that I have, Carly Muir. So Carly, who's pictured 3 places here, did an undergrad project with me that was largely molecular, but as she finished her undergraduate program, she wanted to do something a little more global, a little more ecological. So we turned to a collaboration with Dr. Brian Neff in our department. Brian is a fish ecologist who studies a variety of aspects of fish ecology, including fish mating, but he also does work with the Ontario Federation of Anglers and Hunters, as well as the Ministry of Natural Resources, where he's looking at various aspects of fish ecology, including trying to reintroduce Atlantic salmon back into the Great Lakes. So Carly's project kind of evolved with Brian and me to kind of look at salmon thermal tolerances and performance across temperatures. So Carly's project is largely partly in Brian's lab, where she's looking at sort of ecological aspects, and in my lab, where she works with my other graduate students, Brad and Rachel, pictured over here, to sort of look at molecular and histological aspects of the work. So as I said, so this is sort of ecological data that I hope I am presenting appropriately. So I'm approaching this as a molecular biologist talking about ecology. So when we look at what's happening with salmon today, we see that salmon are struggling. There's increases in global temperature and salmon are not doing well. So there's lots of headlines from various parts of the world that show that salmon are dying. Some of these temperature effects are indirect. There's algal blooms in certain areas that are killing salmon. Some of these effects seem to be directly related to temperature. So there's reports both off the east coast of Canada, so off the coast of Labrador and Newfoundland, where salmon, relatively or healthy salmon, are dying and appears to be due to warm temperature. And also off the west coast, off the coast of Alaska, salmon appear to be dying because of warm temperature. And that's what this picture is from, these salmon that were getting ready to spawn in Alaska. They look healthy. They have healthy healthy eggs in them. They're ready to be swimming upstream, and they died from what appeared to be just hot water, warm water. And when you look at reports of stream temperatures around this date where this report was taken, the stream temperatures in Alaska reached 27 degrees. So it does seem that these salmon are just having trouble dealing with these really high, unusually high temperatures. And that's a problem because I think most people know that as aquatic ectotherms, their physiology is linked to temperature. As temperature increases, their basal metabolic rate increases. So you have sort of a minimal temperature requirement. So as temperature is increasing, your need for oxygen is increasing exponentially. So there's always going to be an increase in minimal requirement for oxygen as temperature increases. So as temperature is increasing, your need for oxygen is increasing. So you have to supply that oxygen. So the body uses a variety of mechanisms to supply oxygen. And in general, the amount of oxygen that can be supplied is greater than what you need. So there's a minimum requirement for oxygen, and you can often deliver more than that. You can deliver a maximum amount of oxygen to tissues. And the difference between this minimum and maximum is called the aerobic scope. So at any given temperature, there's a certain amount of aerobic scope in an organism, and that represents the amount of oxygen basically to survive, to move, and to reproduce, and to hunt. The problem is, as temperature continues to increase, your requirement for oxygen also continues to increase, but your ability to supply that oxygen tends to plateau and then eventually decrease. So if you plot aerobic scope versus temperature, you get a curve like this. So as temperature is increasing, your ability to supply oxygen also increases, and at some point you reach this optimal. So this T-opt is a temperature for a given organism where its oxygen, for its aerobic scope is maximum. So there's a certain temperature, T-optimal, you have maximum aerobic scope. The animal is doing well at that temperature. The problem is, if temperature keeps on increasing, if you go beyond that optimal temperature, your demand for oxygen continues to increase, but you can no longer supply increasing amounts of temperature. So if you keep increasing your need for oxygen, but you cannot supply it, if temperature keeps increasing, there's going to be a point where you cannot meet that oxygen demand, and that temperature is the critical temperature, because you cannot supply oxygen to the animals at that point, and at that point, the animal will perish. So why does this happen? Why does this happen with animals? One hypothesis is something called the oxygen and capacity limited thermal tolerance hypothesis, or the OCLTT. So this hypothesis posits that it's the cardiovascular system that's limiting things. As temperature is increasing, the cardiorespiratory system has to work harder. And it can do so to a limit. So temperature is increasing, the cardiorespiratory system is going to be working harder. It's going to be able to maintain aerobic scope as temperature is rising. However, at some point, the cardiovascular system cannot meet the demands. So beyond the optimal temperature, the cardiovascular system cannot deliver the oxygen needed. And then you get a collapse in aerobic scope, and at some point you're going to not be able to meet the requirements of oxygen delivery, and the animal will perish. Right? So elevating temperatures is a problem for salmon. And when you look at sort of global predictions of what's going to happen, this is from the IPCC from 2013, and this is showing you predicted increases in temperatures by the year 2100. So you can see that most of the world is going to be warmer in the year 2100. And if you look in the areas where salmon are present, you can see that they're going to have to deal with roughly a 4-degree increase in temperature. So the question is, can salmon adapt to a 4-degree change over the next 80 years? And there's a lot of ecological ways that salmon can adapt. There's a lot of sort of slow mechanisms that salmon may be able to change, where they can adapt to these changes over the next 80 years. But my question is there sort of different mechanisms, sort of more short-term mechanisms that salmon can utilize to adapt to these changes in temperature? And one of these mechanisms involves developmental plasticity. So developmental plasticity, as it says here, is the ability of an organism to react to an environmental input at the embryonic and larval stages that results in a change in form. So this has to occur in the embryonic stages. This occurs usually over a short period of time, and it's often irreversible. So there's many examples of developmental plasticity, many that you've heard of. One of the most common that's talked about is sort of the determination of sex ratio in reptiles that's temperature dependent. So in this example, you have turtles and an alligator, and you can see that the sex ratio, the number of males versus females, is temperature dependent, where in general, higher temperatures result in more males. We understand the basics of what's happening here. So you have an embryo, a turtle embryo present. So at a certain stage in development, that turtle embryo has to make a decision, and that decision is based on a temperature cue. So at a certain developmental stage, the embryo looks for a temperature cue, and it uses that temperature cue to make a decision in terms of which developmental pathway it's going to take. So there's plasticity in its development at this stage. So if the temperature is relatively low, it can make the decision, perhaps not the right word, with the low temperature cue, it receives signals to turn on specific genes, and it might take a path down towards oogenesis, towards making ovaries and becoming female. If the temperature is high, it can then turn on different genes that will lead to spermatogenesis and making a male, right? So this developmental plasticity occurs in an early developmental stage. It occurs over a short term, and it's often irreversible. Once you start down the pathway to make ovaries or testes, there's no going back, all right? So developmental plasticity can be profoundly important to certain organisms and their life histories. Question is, do Atlantic salmon demonstrate developmental plasticity? And basically, to kind of steal my thunder and give away the punchline, the answer is yes. I will describe the methodology used to derive this figure later on, but let's just kind of talk about the basics of this figure. This figure is showing that elevated temperatures significantly enhance thermal performance in salmon. So these are groups of fish that were raised at two different temperatures. So there's a control temperature, a low temperature, and a temperature that's elevated by 4 degrees. Okay, so there's fish raised at a green temperature, a low temperature, and fish raised at a elevated temperature, a 4-degree higher temperature, right? These fish were raised through development for a year, and after 1 year, thermal performance was evaluated by measuring various metrics of the heart. And we can see that there are differences between the green fish that were raised at low temperatures and the orange fish that were raised at higher temperatures. So when you looked at their T-opt, so this is their maximal aerobic scope. So fish that were raised at lower temperatures had a T-opt, their maximal aerobic scope was at 15 degrees Celsius roughly. Fish that were raised 4 degrees higher had a different maximum aerobic scope. Their maximum aerobic scope was over 20 degrees Celsius. So raising the fish 4 degrees higher shifted their optimal temperature, shifted their maximal aerobic scope by roughly 4 degrees. So this shows you that you can shift the optimal thermal performance of these fish by raising them at higher temperatures as early embryos. You can not only shift their T-opt to their optimal temperature, but you could also shift their T-crit to their critical temperature, the maximum temperature to which they could survive. So the cold fish, the green fish, had a T-critical temperature of 24 degrees, whereas the warm fish was over 26 degrees. So this shift in the T-crit to the critical upper temperature was not quite 4 degrees, but it was still shifted higher and was still significant. So this sort of take-home message seems to suggest that raising salmon at different temperatures triggers developmental plasticity that allows differences in thermal performance later on in life. So the study is basically, well, how does environmental temperature affect cardiac development to alter heart morphology, heart function, thermal performance in Atlantic salmon? So Carly looked at sort of 4 parameters of this. She looked at gene expression, heart morphology, cardiac function, some of which I just showed you, and some cardiac proteomics. So let's talk about cardiac gene expression first. All right, so to talk about cardiac gene expression, we have to talk about cardiogenesis first. How do you make a heart? So heart development is common in many organisms, including in insects, and it's very well conserved. It's evolutionary conserved. A lot of the genes that are involved in forming this dorsal vessel, which is the heart in flies, are the same genes that are involved in making a heart in us. So there are primordial sort of structures that are formed in all hearts. One of them is the structure called the heart field. So the heart field is basically an area where there's 2 groups of cells that are lying on opposite side of the dorsal midline of the embryo. So you have cells on one side of the embryo, cells on the other side of the embryo, making a heart field. Those cells then come together to form a heart tube. So these structures, this process of a heart field, you have these 2 groups of cells that will migrate together to form a heart tube. That occurs in insects, that occurs in all animals that make hearts, including all vertebrates. So whether you're making a 2-chamber heart in a fish, a 3-chamber heart in a frog, a 4-chamber heart in a bird, which is slightly different than a 4-chamber heart that you find in mammals, all of these processes of cardiogenesis all involve the presence of a heart field, involve the presence of 2 areas of cells that make a field between them, if you like. And these groups of cells then come together to make a heart tube. So these processes are conserved. After the heart field and the heart tube, the heart begins to loop, and then the different looping results in the different chambers of the heart, and afterwards the heart starts to beat. So we're interested in trying to understand what's happening with these early cardiogenesis events. If you raise fish, if you raise any animal, but in this case it's fish, if you're raising fish at different temperatures, are you altering these heart fields and these heart tubes during early development? Okay, are you altering the genes that are responsible for defining the heart field and the heart tube? So this is the experimental setup that Carly did. We obtained salmon embryos from the Normandale hatchery run by the Ministry of Natural Resources that's found in Normandale, which is just south of London. And these fish were raised according to 3 temperature regimes. So the fish are Atlantic salmon that come from the LaHave River, which is in New Brunswick. So the rearing temperature regime used by the Ministry of Natural Resources basically mimics the temperatures of the LaHave River from which these fish come from. So originally you start off with a low temperature, that's roughly the temperature that was present at spawning. Then as seasonal changes increase the temperature, the rearing temperature is increased by the MNR to mimic the temperature increases in the LaHave River. So the control temperature, the 0-degree temperature isn't 0 degrees in terms of absolute temperature, it is unchanged from the MNR regime that's used to raise LaHave River fish. So you have the control regime temperature, and then you have that temperature that's increased by 4 degrees at each stage, or increased by 8 degrees at each stage. So you have control fish sort of at a normal rearing temperature, and fish that are raised at 4 degrees higher at every stage, or 8 degrees higher at every stage. So what we want to do is look at the genes that are expressed during the time where you're forming these important heart structures, the time that you're forming the heart fields, the time that you're forming the heart tube. The problem is, of course, that if you raise fish at different temperatures, they're going to develop at different rates. So to control for that, we use degree days. So degree days is basically just the temperature times the number of days. So 90-degree days is the time where the heart fields are forming. 120-degree days is the time where you're forming the heart tube. 150-degree days is when the heart is looping. And then 180-degree days is roughly when the heart starts to beat. Right, so we're interested to know, if you raise fish at these different temperatures, is there differences in gene expression in important cardiac genes in these fish? And the key cardiac genes that we were looking for were genes that are known to be conserved through evolution in all hearts. And 2 of the most important ones are a gene called NKX2.5, it's also called Tin Man, and another gene called GATA4. So these genes are crucial to forming heart tubes in flies. They're also crucial to forming hearts in mammals. In mammals, they have much more complex functions. If you look for NKX2.5 in here, you can see that NKX2.5 is involved in regulating many other aspects of heart development. We are interested in NKX2.5's functions in early embryogenesis and likewise with GATA4. So we looked at NKX2.5 because it's known to play a role in heart tube formation. GATA4 plays a role in heart field fusion. And there's another important gene called MEF2C. So we looked at the changes in expression of these genes in these embryos that were raised in different temperatures. Was there any differences? There was. So we're looking at changes using PCR of the levels of these transcripts at different stages. So again, the 90-degree days is where you have the heart fields, 120-degree days is the heart tube, 150 is looping, and then heart beating. I think you can see that the greatest change of these genes occurs sort of early in embryogenesis at the time that you're forming the heart field and the heart tube. So it does look like raising these embryos above 4 degrees, whether it's, sorry, above 0 degrees, if you raise the bit 4 degrees higher or 8 degrees higher, you're changing the expression of these important genes, presumably in the heart field and in the heart tube. So what I would have liked to do, what I would have liked Carly to do at this point, would have been to actually do in situ hybridization to look at differences in the domains of expression of these genes. Is the size of the heart field different? Is the size or shape of the heart tube different? But despite the fact that I've done hundreds, if not thousands of in situs on frogs and fish and flies and mice, Carly and I could not do in situs on these salmon embryos. The salmon chorion, the salmon yolk, these embryos were just sort of impermeable to in situ hybridization, at least by the techniques that we tried to use. So an important aspect of trying to understand, is there differences early on in embryogenesis in terms of the heart field and the heart tube, I cannot address. I hope there is, I think there is. There's certainly differences in the genes that are expressed. One sort of tangential way we can kind of try to address what's happening with these genes is kind of looking at other studies involving NKX 2.5. So this is going to start the first part of my hand waving of my data, where I'm kind of going, this is exciting, but really it may mean nothing. So there are many studies that look at cardiac differentiation in vitro and in vivo, where people have taken different model systems and either knocked down NKX 2.5 or overexpress NKX 2.5. So in this studies, they're looking at cardiomyocytes and they're looking at their differentiation in the absence of NKX 2.5. And there's hundreds of genes that are downregulated because of the absence of NKX 2.5 and other genes that are upregulated. So there's many studies that we can look at to try to predict, all right, our embryos have more NKX 2.5, what could they be doing, because there's a difference in NKX 2.5. So we can sort of try to look at some of the transcriptional networks that those changes in NKX 2.5 could be triggering. Why do we want to know that? Well, we want to know whether there's a resultant change in heart morphology. We have more NKX 2.5, we have more GATA4 at these critical stages. We can't do in situ to look at, are there changes in the heart field or changes in the heart tube, but maybe we could look at heart morphology later in 1-year-old fish to see, are there changes in heart morphology that might be associated with these changes in NKX 2.5 and perhaps those associated changes with those downstream genes. Okay, so similar setup to looking at the gene expression. Here I have to point out that the 8-degree fish are gone. Raising fish 8 degrees higher than their sort of native temperature regime is not viable for more than probably 200-degree days. So the 8-degree fish do not survive to a point where we could actually test cardiac function or look at heart morphology. So all the further studies that we're doing looking at heart morphology, heart function, and proteomics was using just the control temperature regime and the +4-degree fish, so fish that are 4 degrees above the control temperature regime. So here we've raised the fish according to these two regimes and raised them to the par stage, which is about 1 year old. And then we are looking for initially changes in heart morphology. All right, so the 4-degree fish I showed to you have an increase in thermal performance. They have a higher T-op. They have an increase in thermal performance. So are these fish going to have morphological changes in their heart associated with these changes in thermal performance? Have other people looked at this sort of tangentially? So people have looked at sort of changes that are associated with aerobic performance in salmon. So other studies have looked at relative ventricular mass and shown that it's positively correlated with CT max in Atlantic salmon, so maximum heart rate. So there's a relationship between maximum heart rate and ventricular mass. So is there a difference in the size of our ventricles in our fish? So we did histology to take a look. So this is a section through the heart of a salmon par, about a 1 year old fish. This large structure at the top is the atrium. The atrium pumps into the ventricle. The ventricle then pumps out through this structure called the bulbus arteriosus that goes out towards the gill. So the ventricle area was taken, and when corrected for body mass, we looked at relative ventricular area between the two treatment groups, the 0-degree fish and the +4-degree fish, and found there was no difference. So there's no difference in the relative ventricular area of these fish. The previous study I just mentioned looked at ventricle mass, not area. So we also looked at ventricle mass, and there was no difference in ventricle mass also. So again, the 4-degree fish have an increase in thermal performance, but that's not due to a size, differences in ventricular mass or area. So what other differences could they be due to? So other studies have suggested that ventricular roundness is bad for cardiac output. So you do not want a round heart, you want a pyramidal heart. You want a heart that's pointy, sort of like the Valentine's heart that you draw that has a little pointy bottom. That's because that facilitates best ventricular contraction and ejection of volume. So you do not want a round heart, you want a pyramidal heart. You want a heart that's pointy at the bottom. Do the fish that we raised at the different temperatures have differences in roundness? The simplest way to measure roundness is to do a length versus width measurement. When you compare the length versus width ratio of the hearts from our 2 treatment groups, of the cold and warm fish, there is no difference in ventricular roundness. So the differences we see in performance is not due to differences in shape. What else could it be due to? Well, other studies have suggested that myocardial thickness provides aerobic benefits. So this is a picture from the publication here that's showing a section of a heart. So again, you have the atrium leading into the ventricle that flows out into this bulbus. The ventricle, here you can see nicely, has this thicker outer layer. That's the compact myocardium. That is the main contractile muscle in the ventricle that provides most of the force during contraction. So do our fish that are raised in different temperatures have different myocardial thicknesses? And the answer is yes, they do. So when you measure the compact myocardium and sort of present it as a percentage of the total size of the ventricle, you see that there's an increase in compact myocardium in the 4-degree fish. So potentially this increase in myocardium helps these fish function better at higher temperatures. Having said that, I should kind of note that bigger is not always better when it comes to myocardium. There is a Goldilocks zone that's sort of ideal. You can't have too much compact myocardium because heart contraction not only depends on the muscle contracting, but also relaxing and then refilling. If your compact myocardium is too thick, you cannot contract fast enough and you cannot relax. You don't have enough elasticity to expand enough. So there is physiological thickening of the myocardium. So that's what happens with athletes. Athletes that work out, they have a thicker myocardium that's physiologically beneficial. So physiological thickening of the myocardium is beneficial versus pathological thickening of the myocardium. There's many heart diseases that are associated with pathological thickening of the myocardium, basically because the myocardium does not function properly if it is too thick. So at this stage, we can't say if this is physiological thickening, which is beneficial, or pathological thickening, which is not beneficial. The simplest way to tell would be just to let these fish grow to an older age. And that's because pathological thickening of the myocardium tends to get progressively worse. So if with time, these fish, the cardiac function of these fish declined, then we know that this thickening is actually pathological and not necessarily beneficial. I don't believe that that's the case. I believe this is physiological thickening, and this is a beneficial thickening of the myocardium, that this myocardium is in part what's helping these hearts perform better at higher temperatures. All right, so for this first part, we looked at a 4-degree increase in developmental temperature and saw that it's not associated with changes in ventricular size, not associated with changes in ventricular shape, but it is associated with a change in myocardium. Okay, so is this change in myocardium, is these changes in this heart associated with changes in cardiac function? Okay, so we have NKX 2.5 and GATA4, they are different in early embryos. You have differences in myocardium that may or may not be related to NKX 2.5. Differences, now we get to cardiac function. So Carly was quite clever and worked really hard for this part, where she took a mouse Doppler system that's designed to measure heart rate in mice, and she adapted it for use in salmon. So Carly developed this relatively simple system where it involved a pipe that she could immobilize fish in. She used lead weights to keep the fish submerged. The fish have a tube in its mouth, so it could be ram ventilated and receive oxygen. Water is circulating through this system, so you could change temperatures. So what Carly could do was take these fish, anesthetize them, pharmacologically stimulate their heart rate so it's at maximum, and then slowly increase temperature, in most cases 1 degree Celsius every 6 minutes, and then measure maximum heart rate as that temperature increased. So you're measuring the maximum heart rate using this Doppler flow velocity system. And what this does is it produced a spectrograph that showed you sort of the changes in velocity that are occurring inside the heart. Okay, so let's kind of look at more how that's working and what the heck is going on here, right? So actually, let me back up a bit. When you place the probe in between the two pectoral fins of the fish, you're basically placing the probe directly opposite the atrium. So the probe is facing right where the atrium is emptying into the ventricle. So you're measuring what's passing through the AV valve, the atrial ventricular valve. So the probe is placed to measure flow from the atrium into the ventricle. Right, so Doppler works by emitting a wave. It emits a wave of a certain frequency. So this Doppler probe is emitting energy waves. Those energy waves will then hit items and then bounce back. And the Doppler probe is measuring these reflected waves. What's interesting is what happens when there's something moving towards the probe. So if there's material moving towards the probe, it will shift the frequency of that wave. So if the Doppler probe is sending out a wave of a certain frequency and something is moving towards it, there'll be a shift in frequency, and that's what the Doppler probe reports on this spectrograph. Okay, that shift in frequency is actually a change in velocity because you're changing that frequency proportional to the velocity of the material coming towards it. The same thing as a radar gun. When the cop is standing at the side of the highway pointing a radar gun at you as you're moving towards it, you're Doppler shifting the signal from that radar gun, and they can tell how fast you're going. Okay, so if you use a Doppler probe with these hearts, you get a repeating pattern that represents the different parts of the heart as they beat. And the easiest thing you could do is place beat markers down, and using beat markers, you can easily detect heart rate. So the simplest measure using Doppler is heart rate. And it is using this heart rate that you can then generate the data that I showed you earlier, where you can determine optimal temperatures and critical temperatures of these fish using heart rate. It's a little more complicated than that, and I can explain to you afterwards during questions if you want to know how it is. But using this, we can demonstrate that there's a shift in thermal performance in the 4-degree fish. The 4-degree fish perform better at higher temperature than the 0-degree fish. The question then is, well, how do you know this is developmental plasticity? You're raising these fish over 1 year at this given temperature, and you told us that developmental plasticity is this short-term event that happens early in development. Well, to address that, what Carly did was she took these embryos and she switched their temperatures to see if you would get a reversion or an acclimation of the temperatures. So she took fish that were raised for 1 year at 4 degrees and shifted them up by 4 degrees. And she took the fish that were raised at 4 degrees for 1 year and cooled them down to the colder temperature. So she acclimated these fish for 3 months at these new temperatures and then asked, all right, what happens to cardiac performance? If this was due to developmental plasticity, then the 4-degree fish should still have an advantage. And indeed, that is what you still see. So if you take 0-degree fish and after a while try to acclimate them 4 degrees warmer, it doesn't matter. These 0-degree fish, if you warm them up later, they're not going to have improved heart function. They did not have that developmental switch. They stay 0-degree fish. They are not going to improve the function. However, if you take the fish that were raised at 4 degrees and then cool them down, they still maintain that advantage. There's still a difference between them and the control fish. So the 4-degree fish did seem to undergo some sort of developmental switch early in development, such that their hearts, even if you warm them up later, their hearts are still performing better thermally than fish that were raised at 4 degrees. So using just temperature, we can get a lot about the function of the heart, suggesting that there is thermal plasticity and there is differences in these hearts of fish raised at 4 degrees. All right, so using just heartbeat, we can, heart rate, we can determine differences in these hearts. What else can you get from these spectrographs of these fish? Okay, so you can get heart rate. And as I mentioned, the measurement of this probe is blood that's flowing from the atrium into the ventricle. So if you look at what's happening during the heartbeat, you also have ventricular outflow. So during ventricular systole, that's when the ventricle contracts and pumps blood into the bulbus, out to the gills in this case. So when the ventricle contracts, the blood is flowing across the probe. So the signals you see during ventricular outflow are largely sort of echoes of movement of things that are moving across the probe. So they're not necessarily useful to figure out what's going on. However, once the ventricle contracts, the ventricle then slowly fills during a process called the E wave. So during the E wave, there's a little bit of blood that comes from the atrium into the ventricle as the ventricle is relaxing. So this velocity, this shift in frequency in the E wave represents blood that's moving into the ventricle during this passive phase of ventricle filling. Then during contraction, you see this huge increase in velocity as the atrium increases and blood moves from the atrium into the ventricle, right? So the E wave and the A wave represents everything that's moving into the ventricle. You see the velocity of things moving into the ventricle, and you can see that there are also peak velocities. So the APV is the atrial peak velocity. You can measure what is the fastest at any point blood is moving. More importantly, though, you have velocities. You have velocities over time. If you have velocity over time, that's basically distance. So if you take the area under the curve of the E wave and the area under the curve of the A wave, so the area of under the curve of these two waves represents a distance. That is basically the total distance that blood moved from the atrium into the ventricle. Okay, so if you have the total distance, you can use that. All right, so you can use this Doppler system to measure heart rate, which we have, and use it to determine the optimal temperatures and the critical temperatures for heart function. You can use it to determine peak velocities, which I'll talk about in a second. You can also use the area under the curves of the E and the A wave to determine total stroke distance. If you have total stroke distance and you know the area that it's passing through, then you can actually calculate a volume. And we can get that value because we have all the sections and we can measure the size of the AV valve. So if you have a distance and you know an area that distance is moving through, that's basically a volume. So you have the distance the material is moving, you have the area that material is moving through. You can use this to get a calculation of volume. So using the area under the curve, the stroke distance, and the size of the AV valve that we can measure histologically, you can calculate a stroke volume. Using this stroke volume, you can then calculate cardiac output. Cardiac output is basically heart rate times stroke volume. So I do have to kind of admit here that cardiac output is generally a measure of blood leaving the ventricle. So cardiac output is the output of the ventricle. Here, we're using stroke volume, and the stroke volume is the volume of blood entering the ventricle from the atrium. They are not necessarily the same. So there's differences in ejection fraction. So the ventricle can eject different fractions under different beats. However, as a proxy, it's not bad. So we're sort of making the assumption that the amount of blood going into the ventricle is going to be the same as the amount of blood leaving the ventricle over any given period of time. Taking this into account, we can then use cardiac output and determine cardiac output at each temperature. And these are the graphs that you get when you plot cardiac output versus temperature. So you can see that the cold fish versus the warm fish have similar slopes of lines. So the cardiac output for both the fish have similar slopes. They both increase with temperature, and that's not surprising because as temperature is increasing, heart rate is increasing, so the slopes of the lines are increasing for these both these samples. But when you compare these lines and sort of look at their intercepts, you can see that these lines are different from each other. Indeed, if you look at any one time point and do t-tests to look at the output, there is a difference in the cardiac output of the 4-degree fish than the 0-degree fish. So at any temperature, the hearts of the +4 degree fish are pumping more blood per minute. Okay, so there is a difference in the heart functions, is that there does seem to be more volume of blood being pumped by the 4-degree fish than the 0-degree fish. You can break down that cardiac output again into heart rate and stroke volume. So when you plot heart rate, so the green is the 4-degree fish and the orange, sorry, the green is the 0-degree fish and the orange is the 4-degree fish, you can see that the slopes of the changes in heart rate are actually surprisingly similar between the two fish. And interestingly, the maximum heart rate between the 0 and the 4-degree fish is not any different. It's the same maximum heart rate. So it's not heart rate that's causing these differences in cardiac output. It is stroke volume. When you look at stroke volume from the 0-degree fish and the 4-degree fish, you can see that the stroke volume of the 4-degree fish is higher at any given temperature than the 0-degree fish. So the 4-degree fish have an ability to move more blood at any given temperature, right? So exactly why that is unclear. So again, this is the volume of blood moving from the atrium into the ventricle, and we're assuming that that same larger volume is moving out of the ventricle into the bulbus. Another interesting thing you can see is that the peak velocity is different between the two treatment groups, right? So the peak velocity is higher in the 4-degree fish. So this is again the peak velocity of blood moving from the atrium into the ventricle. So this suggests that the atrial function in the 4-degree fish is a little bit different than the 0-degree fish. How that translates into changes in velocity, we're not quite sure, but there certainly is a difference. Okay, so we saw that this 4-degree increase in developmental temperature is associated with mass-specific cardiac output. This isn't due to a change in heart rate. It appears to be due to a change in stroke volume. And there's also an associated change in velocity that may or may not be related to these parameters. So you have these fish raised at different temperatures that have differences in early gene expression, things such as NKX 2.5, that result in differences in heart morphology, different than myocardium, that result in differences in stroke volume. Okay, so these hearts do appear to be different. Can we kind of detect these differences in other ways? Can we use cardiac proteomics to figure out, all right, are there differences in proteins in these hearts associated with these different functions? And this is where a lot of the hand waving is going to start again, where I'm kind of guessing things and saying, this is really great and not quite sure what it means yet. But what we did was we took the ventricles from the 0-degree fish and the 4-degree fish, isolated proteins from these ventricles, and sent them off to Toronto for mass spec. And what you get back is peptide counts. You can take those peptide counts and put them into software called MSQ-ROB, which takes these peptide intensity counts and can predict differences between treatments at a given false discovery rate. So using, in this example, a false discovery rate of 5%, MSQ-ROB suggested that there's 77 differentially expressed proteins between the two treatment groups. There's 43 proteins that are upregulated in the 4-degree fish and 34 proteins that are downregulated in the 4-degree fish. So there's a lot of data here, and there's a lot of things we can do with it. And again, a lot of it is a bit of hand waving. Certainly when you look at these genes, there's a lot of genes that make sense. So there's tropomyosin and troponin and myosin binding proteins. There's a lot of genes whose expression is different, something that you would expect if these hearts are functioning different, but it doesn't necessarily tell you that these hearts are functioning better because of these proteins. Another problem is, and I should have shown you a bigger Excel file than this, is that the salmon genome is not well annotated. So for a lot of the peptides that were identified by mass spec, there is no known protein. So there's a lot of unknown peptides in there that makes sort of analysis a little more difficult. You can take these proteins and then also try to do different sorts of analysis with them, again, in the context that most of these analyses are related to mammalian hearts and mammalian functions. However, you can take these proteins and put them in through a variety of analyses, including analysis of gene ontology enrichment. So I know you can't see the writing here. It's sort of reflected in the list here. So what you can do is you take these 77 genes and you put in the databases, and ask, all right, is there enrichment of these genes in any given known molecular function? So this output tells you that these genes that we have are known to be working together in actin binding and cation binding and calcium binding. So there is a statistically significant increase in genes, for example, that are known to be involved in calcium ion binding. Potentially that's crucial to changing heart dynamics and heart function. So there's lots of promising avenues of those genes and how we can interpret what they're doing and how they might be related to the differences we see in heart functions. And sort of the last bit of hand waving here, we can also take those genes and try to compare them to a lot of these analyses that have been done in human hearts. As I mentioned, differences in myocardium, particularly in pathological thickening of the myocardium. There's tons of studies that have looked at genes that are up and down regulated in human conditions of problems with the myocardium, with the thickening of the myocardia. So we can, again, use our genes to look at, to see, are our genes upregulated in other conditions where there are thickenings of the myocardium? Is there something we can glean from this information? I know I look like I'm doing jazz hands the amount of time I'm waving my hands, but it's a lot of possibilities that we are looking into, and I'm hoping something useful will come out of it. I think it is intriguing that there are differences, and a lot of these differences are related to sort of other studies in humans and in other animals. All right, so kind of summarizing the whole thing. So we have a 4-degree increase in developmental temperature is associated with improved thermal tolerance, differential expression of cardiogenesis genes, differences in heart morphology. So you have an increased proportion of the compact myocardium. There is no increase in cardiac output. Sorry, there is an increase in cardiac output, but it's not due to differences in rate. It's due to differences in stroke volume and perhaps to stroke velocity. And there are differences in the proteins of these heart tissues. There's 77 of them. Are these differences related to things such as NKX 2.5? Are these differences related to cardiac hypertrophies that are present in humans? We are looking into this. I think it's sort of a beginnings of a nice story. I think, though, together it kind of shows that there is evidence for temperature-based thermal developmental plasticity in salmon that's impacting thermal performance and heart function and cardiorespiratory morphology. This is consistent with the OCLTT method, the OCLTT hypothesis that suggests that it is the cardiorespiratory system that is limiting sort of these changes. And this data can perhaps be used by Brian and other sort of fish ecologists to kind of determine the best way to rear fish at what temperatures and to look for what sort of aspects of myocardial thickness when they're rearing fish to do different sort of reintroduction of fish into different places, such as in Lake Ontario. And with that, I will thank you and take questions. Should I stop sharing my screen?
You can keep sharing it. That's fine.
All right, sure.
So I guess we'll all give Dr. Damjanovski a nice virtual clap for a great talk. And we'll open the floor to questions. So feel free to input your questions into the chat feature, or you can unmute yourself and ask a question.
I have a question to start.
Uh-huh.
So I think you got it in the beginning of your presentation. You kind of looped back to it at the end about this thickening, was it myocardial tissue?
Yep.
And then in your proteomics experiments, you're kind of starting to look at whether or not that is related to like if that's some sort of like pathogenic response or if that's just a physiological response.
That's correct.
Do you have any insight yet, or is that something you just are still doing experiments on to see?
Yeah, a lot of the proteomic stuff was literally the week before last. So
Okay.
we're just putting that together, trying to, first of all, sort of validate a lot of the stuff. That first Excel file that I showed you where the gene ontology numbers, like we have to manually find all our genes. So the one database will tell you this is salmon gene, whatever, and then you have to kind of verify that indeed that is that gene. So you have to go to other databases and properly annotate this stuff. So it's taking a long time to annotate all these genes. So at some point, yes, I will find a nice representation of, if we can, of sort of physiological thickening of the myocardium and look at genes that others have reported that are increased in physiological thickening and see if they're also upregulated in our data. But we haven't really looked at that yet.
Right. Okay. Interesting. Thank
Yeah,
you.
and on the topic of proteomics, I wonder if I could ask a question. I think it was really enlightening to start to get some sense of what's happening at the protein level and what proteins might be present in differing amounts. Now, I noticed at least one voltage gated channel, and I think you alluded to calcium signaling potentially being involved. I mean, do you think that there's any signaling that's maybe enhanced in these fish, signaling via gap junctions between the different cardiac muscle cells? I mean, do you have any idea on what might be happening? What ions might be transported?
That would be, yeah. So learning about, the regulation of heart contraction, and it's so complex between the pacemakers, between gap junctions, between hormonal regulation of things. We have been looking at things. There's something called atrial neural peptide. I think Carly's on here. So there's a peptide that's often regulated with regulating heartbeat. So we looked at that. Are the differences in these fish? So no, there isn't. So yes, we are aware of sort of the different mechanisms. We're trying to look into it, except that that's not my strong suit. So I'm trying to find other people. So if you notice on the acknowledgement slide, there's a faculty member named Tom Drysdale who does heart development. So I'm sort of working with him to try to figure out, all right, which one of these things is going to be involved in perhaps regulating. Is it, the gap junctions or channels or whatever? And I don't have an answer to that yet.
And maybe one quick follow-up question is, what do you think is happening when you bump it up to 8 degrees? Do you think the myocardium becomes just too thick? Like, do you think there's a threshold?
I think, no, I think it's too long at 8 degrees, I think. So the problem is with that developmental plasticity, it should be over a short temperature, a short time regime. So they were raised at 8 degrees for, months. And I think that's the problem. Perhaps if you raise them at 8 degrees for 1 month versus 8 months would be the key. And then they would survive. They would have a heart that would be developed to a certain extent. And then whatever else that 8 degree increase is doing, perhaps isn't going to be as detrimental. I think the problem is that we kept that 8 degree temperature regime going much longer than needed. And in general, I mean, when we did this, we don't know when is that developmental plasticity going to kick in. In principle, it could be related to heart function, and I have other slides here. The other issue is, of course, gills. So we also want to look at differences in the gills, which are sort of later. So we're also looking at, are these differences in rearing temperatures related to gill function? So there is many sections of gills here that I didn't get to. In general, there is some differences in the gills, but not the ones you'd think. So it's too complicated to explain unless you want me to. So there are differences in the gills. So there are other things that this temperature increase can impact in terms of developmental plasticity. So perhaps keeping it 8 degrees is just impacting too many things over a long period of time. I think it's maybe better if you have an increase that's going to affect heart at this stage, that's fine. If you have it increasing heart and gill and blood chemistry and everything else, then it's just maybe together it just doesn't work. If that answer made sense.
Yeah, awesome. Great talk.
We have a few questions here in the chat, so I can ask.
Sure, I don't even see the chat, but go for it.
Yeah, so we have one of our audience members, Ann McCarthy, said, is there any thought to looking into more wild type strains of Atlantic salmon instead of LaHaves? They are a legacy broodstock in fish culture, but are quite domesticated. Or do you think that would have any impact on the results?
I can't answer that well, just because I'm not quite sure what the differences are between them. So I know this stuff, when we started, included several different salmon strains. So Sebago comes to mind. So there was different salmon strains that were sort of initially started. And I think these were just the easiest for us to grow in our facility. So yeah, I think there is probably going to be differences between them. And even between Chinook and these guys, I think I sort of surprised how many different types of salmon there were when we first started doing this. So I'm sure that is a consideration, and I'm sure that, or potentially the results we have might be more specific to LaHave, specifically the time point at which that developmental event is happening. So the LaHave River salmon probably have heart development occurring at a specific time point, and you got to catch that time point where you're increasing temperatures, and other salmon may have sort of that different time point for that developmental plasticity event.
Okay, thank you. We have a few more here. So one of our faculty members, Dr. Knoll, asked, I think this was when Rob was talking, one of his previous questions. Similarly, is there any evidence that there is natural variation in any of these characteristics as a function of ambient water temperature for embryos that natural selection could act upon?
So yes, there was a part of my introduction which I removed, which does suggest that there is quite a bit of plasticity in terms of the adaptability of these salmon. So I'm not sure if I can answer that question properly, but I would say that other studies, for example, there's another study by Brian Neff's group. They were looking at Chinook salmon. So they were raising Chinook salmon at different temperatures, and they weren't looking for developmental plasticity per se. But when they looked at the salmon, siblings that were raised at a higher temperature displayed higher optimals that were correlated with increased heart rate. So the Chinook are kind of doing the same thing, but not exactly the same thing. And they weren't necessarily looking at developmental plasticity for this study, but it does appear that there is variation within populations, between populations, that is going to impact things. But I am not the one to speak about the different types of salmon, unfortunately.
I have one more question. I'm not a developmental biologist by any means, but physiologist even. But do you anticipate that there might be any decreases in fitness of these individuals because of increased heart growth or something like that? And maybe is that a consideration that you'd make further down the line if there were considerations about introducing these raised individuals into wild populations?
Of course. So yeah, I was trying to allude to that when I mentioned that, yes, if you raise them at a higher temperature, you get an increase in myocardium. And that seems good, at least at this stage, at the par stage when you're testing them. But if you're testing them, much later when they're out there and they have to compete in things, perhaps it's not an advantage. So I think you need a lot more to figure out, is this really providing an advantage? To me, it's sort of a stopgap. If you need, a mechanism where you can assure that you can rear salmon at a slightly different temperature and then introduce them into an environment where the temperature is elevated, and these salmon have a better chance to survive at this elevated temperature. That's sort of the first part. At least you know that they'll do okay with the temperature. In terms of other aspects of their fitness, whether they're competitive or something, yeah, there could be huge issues with it. But I'm just sort of, at least that part of it, that it's going to deal with that temperature increase over the short term better, is sort of what I'm looking at the moment. At least that's a bonus to some extent.
And one more quick question from one of our students. She was wondering if the OCLTT hypothesis applies to humans as well.
Well, I think it's just for ectotherms. So it is because your basal metabolic rate is increasing with temperature, and I do not believe our basal metabolic rate increases with temperature as much. I'm sure it does to some extent. So I don't think so. Having said that, there is certain amount of plasticity in humans as well. The best example is twin studies. If you separate twins early in development and under different environmental conditions, they do display differences. So it's not like there isn't plasticity in most things. You do use different genes under different environmental conditions. So that's not unusual to turn on different genes because of environmental cues, not necessarily because of sort of endemic maternal determinants or other sort of transcription factors.
Okay, I think that's all of our chat questions. Yeah, if anyone else, does anyone else have a last question to ask? Oh, all right. Well, thank you so much for spending today with us, and thank you so much for the excellent seminar. We appreciate your time.
Yeah, thanks, Sash.
Thanks. No, nice to see people and sort of meet people, and hopefully we'll chat with others soon. See y'all.
Great. Bye.